Hi guys, welcome to part 2 of speciation. This is where we ended last time, with the whales returning to the ocean. And how do we know this? And is there evidence for this? What kind of evidence do we look for? So look at this whale skeleton, and we actually can see some anatomical structures that are vestigial. Remember the word? That are leftover evolutionary anatomy part. And where do you see that? This is vestigial hips. This is where some of these limbs would have been, and there are still some parts of bones left behind there. Super interesting. This is at the Seymour Center. If you haven't gone, if you're a local and haven't gone, you should definitely go see the blue whale that they have there, and they've preserved all the bones. It's beautiful. And also there, hanging, they have right there and there, you're able to see those vestigial hip bones. It's so much fun to look at humpbacks in our bay. I don't know if you've seen recently, but they've been out, and I thought I'd classify them. So kingdom Animalia, phylum Chordata, they have a cord and backbone. Class Mammalia, order Cetacea, family Balanopteridae, Megaptera is the genus, and the species is Novangeli. So those are the guys that we can see here when they're cruising by, that is so majestic and fantastic. Just as a reminder, these are some of the classifications. We started with domain, dumb, kids playing cards on freeways get smushed. We have that of the human, where we compare ourselves here to an ostrich. So we both are in the same domain, in the same kingdom, in the same phylum, but then we separate out. We go into the class Mammalia, and they go into the class Aves. And then order we're primates and Strathyscenoformes. And so it's going to vary as it goes down here. But this is the ostrich, is a Struthiocamelus, is a genus and species. You don't have to memorize this. I want you to know the order, though, like you did on the first test. This is a video on Crash Course in Biology, and it's on speciation, and I'd like you to see it. It's posted in the external video section of my Canvas. The last section here in the chapter is when they have some research paper, and it's been summarized for you. And this one's called Investigating Life, Birds Do It, Bees Do It. It's very interesting. It's about two closely related plant species, and the genus is Mimulus. And this was the basis for a study on the causes of speciation. In other words, when will something speciate? How does it speciate and become two different species? You know, how does it even happen when you don't have a barrier, when they're sitting side by side? So this was a nice little study that tried to look at that, and they used this Mimulus plant that you've probably seen around here. And I think I have a Noticing Nature that has an orange one. So this is monkey flower, and this one is a wild type, and it's quite purple. You know, it looks purplish. And this is another wild type here of a different species. It's Mimulus cardinalis, and also monkey flower. But this one's more orange. So this more towards the purple, this towards the orange. And it turns out that this one's more likely to be pollinated by bees. So bees will visit this flower, and hummingbirds will tend to visit more of the cardinalis species. So let's think about how these got so similar, how they got to be separate species. This is just an enlargement for you to see the difference in color. And this is some of the genetics of it, making it purple, and the genetics of this one making it red. So this is the hypothesis. Scientists hypothesized that one mutation changed the flower color. And by changing the flower color, they became different species. Now, how can that be? So if one mutation changed the color, it started the speciation event that separated them and they no longer reproduced. What would make it so that they no longer reproduced? So to test this hypothesis, they use selective breeding, which is, like we do all the time. We want a cucumber that's crunchier and we go selecting for crunchier and crunchier cucumbers and we back cross it to crunchy cucumber until we get a really crunchy one. So they did this, but what they did was they selectively bred each species so that it got closer to the color of the other one. So when I started with one that was very purple, I every time one was a little more orange, I bred it again to the orange one and back again to one that was a little more orange until after various generations, you get one that's a little more orange. And you do the same with the other wild type, with the Cardinals that used to be quite orange and it's shifting over to being more pink because you've crossbred it over and over again, always selecting for the more pink one. So what happens? Well, if we had bees visiting the Lewisy way more, and the Cardinals being visited only by hummers, what happened after they start to look kind of alike? The new varieties had different frequencies of pollinator visits. So whereas we had many bumblebees on the wild type of the pink one, and very few hummingbirds. Then when you selectively breed it and make it look more orange, this number drops. Look at this number as compared to this one. Drops dramatically, and I start having way more of the hummingbirds. Well, that's what happened with the Lewisy. What about the Cardinals? In the Cardinals, we had very few bumblebees visiting, right? This is the bumblebee column, and way more hummers. And we increase the visitation by bumblebees and diminish that by the hummers. It doesn't mean that it totally shifts, but you have way more cross-pollination once they start to look more alike. These are the numbers again, just bigger to make sure that you can see them. Gone from 15 down to 2, from 0.02 up to 1.4. And this is in visits per flower per hour. Okay, so what is this telling us? It's suggesting that a mutation in a gene that affected flower color, so all you needed was one of these way back when to be affected by a mutation that changed the color, and it caused this shift in pollinator preference. And so if you have bees only going to one and hummers only going to the other one, then what happens is those get separated reproductively. And it isolates those two flowers, given that they're not visited by the same pollinators. So as little as one mutation, one little thing that changes the color may turn these flowers into different species, which is really cool to think. It isn't 15 different things that have to happen to it. You know, grow taller and change color and maybe make this opening broader or shallower. No, all it had to do was change one thing, which was coloration, for it to separate sympatrically, right? They're in the same fields maybe, into two different species. So that was the result of that study. One last thing I want you to try and do here is to draw a cladogram using fruits as the organisms with something in common and reflecting relatedness. So what does that mean? If I told you that you had to build your own cladogram like the ones I've shown you, but this time you're going to use a walnut, a plum, a peach, a tomato, a watermelon, an orange, and a lemon, and you have to construct, some sort of branching that goes like this and maybe like this, and in that branching you would have to be able to put one of these on the end of these branches giving an idea of which were the ones that were more closely related and which were the ones that were more distantly related. And I gave those to you and said, here, do it. Where would you go from here? So you can use this. And the hard thing about this is deciding which are the structures that are so important that makes them different. So I'm giving that to you because that was a hard thing to come up with. So someone else did this for you and they said, well, you'll want to check whether seeds are present and in which have seeds and which don't. And that's probably a characteristic that's going to tell some apart. What other characteristics? Well, how about you check which are the ones that are soft inside? Some are not. What about checking about, seed size? Some of them will have small seeds. Some of them won't. Some of them will have a big stone like a peach. And then some will have a really thick skin like an orange maybe, and not like a plum. Right? So that's going to tell some of these fruits apart. And then some are going to be segmented, which means, it's got pieces like this. Oh my gosh. Like an orange. That doesn't look very good. So they have slices, essentially. They have their own slices, which means segmented. So what you need to do is, at the end here is going to be a fruit, and another, and another, and another fruit in this cladogram. And I want you to be able to do that. I'm not going to give it away right now. Maybe at the beginning of the next lecture, I'll show you what the solution was, but I think you can figure that out by using these characteristics. And that's what researchers, scientists do. They get a bunch of moths and butterflies, and then they have to decide, okay, what are we going to look at? We're going to look at the antennae. We're going to look at the coloration. We're going to look at which one, the ones that have the longest wings and the shortest wings, which are the ones that have the longest tongue and the shorter tongue, and put them together in a way that reflects common ancestry and seeing which are the ones that are more closely related. So this is sort of a very simplistic exercise here with a fruit. And it doesn't probably reflect ancestry. It's just an exercise for you to see how this is done. So go to it, see if you can do that. That's the last slide in my lecture. Thanks for watching. Bye.